Chromosome. We know DNA carry genes, and these genes they are responsible for carrying information from one generation to the another generation. Actually, genes are responsible for producing different kinds of proteins, and these proteins they make up an organism as per the instructions given by the genetic material. What type of cell should be produced, in what quantity each protein it should be produced. All these things they are determined by the genes. Genome of eukaryotes is divided into chromosomes. How genes are typically arranged on each chromosome is also very specific. See case of human being. Each human cell contains about 2 meters of DNA if stretched end to end. Yet the nucleus of human cell, which contains the DNA, is only about 6 micrometer in diameter. This is geometrically equivalent to packaging 40 kilometer of extremely fine thread into a small tennis ball. This is complex task, packaging of DNA. No doubt it is a complex task, but is accomplished by specialized proteins which bind to and fold the DNA, generates series of coils and loops that provide increasingly higher level of organization, that is coiling and supercoiling. In this figure we can see linear form of DNA. Actually then its circular form, and here coiling and supercoiling of the DNA thread. And in doing so, or while making coiling and supercoiling, or making the DNA compact, DNA is prevented from becoming unmanageable tangle. Actually, very wisely, DNA is tightly folded and compacted in a way that allows it to easily become available to many enzymes in the cell that replicate it, that repair it, and use its genes to produce proteins. How DNA packaging occur in prokaryotic DNA? So this is explained in this sketch. So venous bacteria, they are prokaryotic. They carry their genes in single DNA molecule, which is double stranded, and circular. This DNA is associated with proteins, thus perform packaging and condensation of DNA, but they are different from proteins that perform these functions in eukaryotes. Bacterial chromosome, it does not have the same structure as eukaryotic chromosome. Now, packaging of eukaryotic DNA into a set of chromosome. In case of prokaryotes, what happens that we have seen here. Now related with the packaging of DNA in case of eukaryote, where particular set of chromosome is formed. So human cell, it contains over 2 meters of DNA packaged into a nucleus of 5 to 20 micrometer in diameter. To achieve this, DNA is packaged in highly ordered process. This allows condensation of DNA, but still enables replication and transcription machinery access. Several proteins are involved in the condensation of DNA. Levels of DNA condensation we will see in eukaryotic chromosome. The first level of DNA condensation occur when 146 base pairs of DNA is wrapped around histone proteins named as nucleosome. DNA is wrapped twice around the histone core to form the beads on a string level of packaging and is termed as chromatin. The second level of chromatin condensation is arrangement of nucleosomes into 30 nanometer chromatin fiber, where 6 nucleosomes per turn are found, and this structure is termed as solenoid. So this figure shows how folding or coiling and supercoiling of basic chromatic material to form chromosome. See here you can see chromosome. From this chromatin material, how by the process of coiling and supercoiling, at last chromosome is produced, or DNA is packed in the chromosome. Chromatin fiber is then arranged in loop domains, each approximately 120 kb long. These chromatin loops form rosettes. Each rosette is termed as multi-loop subcomponent, and it comprises of 6 loops. Further, 120 kb of DNA links each rosette. Rosette is attached at its center to the nuclear matrix, and is associated with histone H1. Now final level of chromosome compaction occur prior to cell division in mitosis. Mitotic chromosomes have diameter of 700 nanometer. Rosettes are lined up and attached to central chromosome scaffold on up non-histone acidic proteins. Now we will see types of chromosomes. So on the basis of number of centromeres, chromosome types are there. On the basis of position of centromere, also the types of chromosomes are made. So on the basis of number of centromeres, chromosomes are of different type. So monocentric when there is only one centromere in the chromosome, dicentric with two centromeres, one each in chromatid. Polycentric means more than two centromeres in chromosome. Acentric is another type. It means it don't have centromere. Then diffused or non-located centromeres are distributed throughout the length of chromosome. So like this, on the basis of number of centromeres, some types like monocentric, dicentric, polycentric or acentric are there. On the basis of position of centromere, see in this figure, the position of centromere. Here it is at one end, here somewhere near one end, and like this. So based on position of centromere, the types of chromosomes are telocentric, acrocentric, submetacentric, and metacentric. So telocentric type of chromosome. These are rod-shaped chromosomes with centromere occupying a terminal position. This is its terminal position. Here in this chromosome, one arm is very long and other arm is absent. This is one arm, it is very long and other arm is absent. Second type is acrocentric. These are also rod-shaped chromosomes which is having subterminal centromere. This position of centromere is subterminal. In this chromosome, one arm is very long, this one, and other arm is very small. Third type is submetacentric chromosome. Submetacentric chromosome. So these chromosomes looks like J or L English letter, where centromere slightly away from midpoint. Centromere lies slightly away from midpoint so that two arms are unequal. This and this arm, it becomes unequal. This is submetacentric chromosome. And metacentric chromosome. It looks like V, English V alphabet. Here centromere lies in the middle of chromosome, exactly at the middle, so that two arms are almost equal. So this is what related with the types of chromosome based on position of centromere.